Hey guys, good morning. I'm starting one of the lab for spatial senses. So you can go over them and then you learn about all the labs I have posted. So to go over the lab, we have, as usual, we will go over Open My Lab and Mastery. And I will go over the models, 3D models in the spatial senses and the histology of the spatial senses. So let's go to study area, launch the study area, and then we will go over practice anatomy lab. Then we will select practice anatomy lab, anatomical model, nervous system, spatial sense. Okay, so in a spatial sense, the first structure we're going to see the accessory structure of the eye. So you want to start with the showing levels first, and you can see the general structure of the eye and all the levels. So this is lower part of the orbital cavity bone here, and then you have eye. In the eye, you see this is the lateral portion of the eye. You see lacrimal gland under your eyelid, sorry, the bone above your eyelid. And then there is some muscles. Here is lacrimal sac, the medial area. This is the superior palpebra, which is the superior eyelid, inferior eyelid. And then inside you have the sclera, iris, pupil. And this is some muscles and ligament, lateral palpebral ligament. And these are the superior inferior colliculi. So these are the structures of the eye. We will go these all structure in detail, okay? So let's go to next slide. And let's see the 3 layers of the eyeball. So outermost layer is called sclera, as you know, and then inner tunic is vascular, and the innermost is the sensory neural, which is retina. You cannot see here. The choroid layer interiorly is modified, and we have here ciliary body. In the ciliary body, we have ciliary muscles. Then we have iris. The ciliary muscles is attaching inside to the lens, which they adjust the image to focus on your retina. And this is the hole in the cornea, that is the pupil. Okay. Now, again, internal structure of the eye. So let's hide the label here so we can see it easily. So outermost part here, this is sclera. Then again, choroid. Choroid anteriorly becomes the iris. And around the iris, you have ciliary muscles. And then behind, you see lens here. Behind the lens, you have the very thick vitreous jelly-like fluid, which holds your retina to the wall of the eyeball. That is called vitreous humor. Vitreous humor lies in the posterior segment. So lens is the boundary between the anterior and posterior segment of the eye. You can see all the levels again. Okay. You can see this is the ciliary body. Okay, so this is sensory and vascular tunics of the eye. That means we have removed sclera and looking at the vascular and sensory tunic. Okay, so this is like transverse section of the eye, and you are looking from the top. So this is the outer part, this is the choroid, and then you have the retina. And when choroid and retina are transitioning, that area is called ora serrata here, this like squiggly line, that is ora serrata. So let's go over the choroid. Choroid is anteriorly attached to ciliary body, which holds the ciliary muscles, and ciliary muscles then attach the lens, hold the lens, which attaches to the ciliary body through the ciliary zonules. And then anterior part is iris. Okay. Posterior to the eyeball, you see this is optic nerve. The yellow part is the axons of axion cells, and then optic disc. And then there is central artery and central vein, and this white part is sclera. Here is a small pit on the posterior wall of the retina. That is called macula lutea and fovea centralis. So the larger area is macula lutea, and the point in the center is macula fovea centralis. This is the only area where there are more cones but less rods. Otherwise, all over the retina, there is a lot of rods and very few cones. Fibrous and vascular tunics of the eye again. So you see here, this is the sclera. And all the sclera part, you see they are opaque, light cannot enter. But when you come to anterior part, you see this part, this area particularly, this area is, to see this area, you have to move your pointer. This is transparent, and this transparent area is called cornea. Behind the cornea, you have this hole that is pupil, iris, and this cavity here is called inside the cavity. Cavity from lens up to this cornea is called interior segment, and this area is filled with aqueous humor. Okay, this is the ciliary muscles. Then you have sclera, and you can see some extra ocular muscle, extrinsic eye muscles. There are some nerves, the choroid, and then other nerves, and then optic nerve exiting from here. This is the cut section of posterior part of the retina. So posterior half of the eye, anterior view, and you can see all the layers again. So always keep in mind the outermost layer, choroid, then the retina, and in the retina you have this whitish area that is called optic disc. This is the point where your optic nerve starts. Now, extrinsic muscles of the eye. So eye has two kinds of muscles. One group of muscles which are outside the eyeball, another group of muscles which are inside the eye. So the muscles which are outside the eyeball are called extrinsic eye muscles. They are skeletal muscles. And these muscles are superior rectus, inferior rectus here, lateral rectus, and you have both right and left lateral rectus, superior oblique, and inferior oblique. So there are like 6 muscles. These muscles moves your eye in different direction when they contract. And then iris, pupil, you can see again here. This cavity is orbital cavity. So if you take out this eye, the cavity in your face is orbital cavity. This is the posterior view of the eye, and you can see the muscles. Medial rectus, lateral rectus, superior oblique, inferior rectus, inferior oblique, and then optic nerve. And this is the sheath of optic nerve, which is pretty much dura mater. Now let's start with the ear. So ear is the organ responsible for hearing and balance. Ear has three parts, area. So here, this is the tympanic membrane here, or we call them eardrum. This tympanic membrane is the boundary for the external and middle ear. And this middle ear cavity is called tympanic cavity. You see, tympanic cavity, which is continuous with your pharynx through the pharyngotympanic tube. So it opens into your throat to balance the pressure inside and outside this tympanic membrane. Okay, so external ear has auricle, then you have external acoustic meatus. This external acoustic meatus is also lined by the same skin layer, stratified squamous. So whatever infection you can get into ear, outside ear, you can get into this external acoustic opening or meatus. Then tympanic membrane, from here up to here, it is middle ear cavity. So this is all middle ear. Okay, in the middle ear, you have tympanic cavity. Inside the tympanic cavity, you have three smallest ossicles, malleus, incus, and stapes. Okay, so this is all middle ear cavity, and this all cavity is in your temporal bone. You see this big cavity. Medial to here, the foot process of the stapes, you see, foot process of a stapes is attached to the oval window. And from here, everything inside is inner ear. Another name for inner ear is labyrinth. So inner ear has cochlea, vestibule, and semicircular canals. Cochlea is responsible for hearing. Vestibule and semicircular canals are responsible for balance of your body. So from this inner ear is the sensory organ of the ear. That's why you see vestibulocochlear nerve is exiting from here and going to your temporal lobe of your brain where you hear things. Okay. So inner ear is called labyrinth, and inner ear is like a maze, which is the like twisted tubes inside the bony labyrinth. So the wall of the inner ear is made up of bone of your temporal bone. And inside that there is membranous cavity. Okay, so that's inner ear is like tube within a tube. Okay, so let's see if we cut anywhere on the cochlea or vestibule or semicircular canal, you're gonna cut outer tube, which is made up of bone, and then you're gonna go inside, there is inner tube, which is made up of membrane. Okay, so let's go and see with the levels everything here. See here, there are three semicircular canals, and when you remove the bone, you're going to see semicircular ducts. So bony labyrinth is removed here, and you can see here ducts. Okay, so all structure here is membranous labyrinth. So here, this is lateral semicircular duct, this is posterior semicircular duct, and this is interior semicircular duct. Here is vestibule. Vestibule has two parts: utricle towards the semicircular canal and saccule towards the cochlea. And we talked about all the structure. Okay. So let's see inner ear. Here we have the inner ear with bone. Earlier we saw only duct. Now we have bone. So you see, this is the tympanic membrane. From here outward will external ear, and then inside here you have malleus, incus, and stapes. Stapes has food process like the horse, the stirrup when you ride horse. You put your leg on a stirrup, and a stirrup has foot process. It is attached to the oval window here, which is not shown. Okay, so this is vestibule, semicircular canals, and cochlea. And outer part is bone, and if you cut, then you're going to see inside, you're going to see membrane tube, and there is vestibular and cochlear nerve together called vestibulocochlear nerve. Okay, so let's see if we cut here. Let me show you with so let's see if we cut here, or if we cut here, what are we going to see? It is like a tube inside a tube. So outer tube is bony tube. So first you're going to cut bony tube, then going to see a space, and then you cut membranous tube, and then again you're going to see space. Same thing here too. Okay, so we will see that later. Okay, so these are just the ossicles, three smallest bone in the middle ear. And from lateral to medial, it is MIS, malleus, incus, stapes. And this area is stapes foot process. And can you guys answer me where this foot process is attached to? To the oval window of medial wall of the inner middle ear cavity. Okay, so as I said earlier, let's hide the tube. So if you go and if you cut cochlea, so cochlea is like a snail. It has 3.5 coil circle. And if you cut cochlea in any place, because it is a tube within a tube, yes? So outside you have bony tube, and then inside you have membranous tube here. So in the membranous tube, so here, let me tell you how. So when you cut in cochlea, there are 3 cavities inside the membranous tube. The cavity which is continuous with the oval window is scala vestibule. The cavity which is in the center of membranous labyrinth, scala media, which is in the middle. And the same scala vestibule which is continuous with the round window is scala tympani. And you can understand this. This is a little bit tougher concept, and we have not gone over ear. So you will see that later when we talk about it. So this is scala tympani. This is scala media, which is contains all the receptor organ for hearing. And that receptor organ for hearing is called spiral organ of Corti. So spiral organ of Corti is the organ of hearing, the receptor for hearing. Corti is the name of scientist who discovered this area. That's why we put his name. So this is, now they are separated. So scala media is separated from scala vestibuli by a membrane, and that membrane is called vestibular membrane. And then the membrane between the tympani and the media is basilar membrane. On the basilar membrane, you have spiral organ of Corti, and there are, you see small things here. Those are the longitudinal cells, columnar cells. Those are the receptors. On the receptor, you have tectorial membrane. So 3 membranes you need to remember: basilar membrane, vestibular membrane, tectorial membrane, and basilar membrane. Okay, and these receptor cells have axons which come out and becomes the vestibulocochlear nerve or cochlear nerve. Spiral organ of Corti, highly magnified. You can see we are focusing now on scala media. So you see here, we have basilar membrane, we have supporting cells, and then you have outer hair cells. These hair cells are hearing receptors. When sound comes, they move the fluid inside the scala media, and that moves these hair cells, and then they depolarize, they text the impulse to the brain. Okay, you see the tectorial membrane and the scala media. Now the eye, the nose, this is the sagittal section of your nose. So you are separating your nose in right and left half, and then you are looking here from the side. So this is the roof of ethmoid bone, where there is cribriform palate and olfactory bulb is passing there, and you see olfactory nerve is synapsing in your roof of your nose in the superior nasal concha. When you sniff certain things, it comes here, and these chemical compounds depolarize these nerve fibers and you smell. Okay, so these are the nasal cavity and the smell. The tongue has the, let's see here, the papilla, the valid papilla, fungiform papilla, and there is other papilla which is called circumvallate papilla. These papilla contains the taste buds, which you can taste when you put chemical compound on your. In the posterior part, you can see this is the throat and the uvula, okay, on superior. Okay, so now I'm going to go over the histology of the spatial sense. So let's go to histology, and then nervous tissue. Let's go back. Spatial senses histology. Okay, so in the ear and in the eye, histology is only two things: histology of inner ear and histology of retina. That's you have to focus on. Okay, so cross section of retina here. So retina has from outside, the outermost tunic of the eye is sclera, then you have choroid, and then you have retina. And in retina, last time we did, there is several layers. So inside the retina, we're going to talk about what are the layers of the cells in the retina. Okay, so this is choroid, first three layers of the eyeball, and then the retina. See, sclera, choroid, and retina. Now the magnified view of the retina. So what are the layers of cells in the retina? So again, this is outside. So sclera, the small choroid, and then retina. Retina has several layers. The outermost layer of the retina is nuclei of rods and cones. Then you have bipolar nuclei, bipolar neuron cells nuclei. Then you have nuclei of ganglion cells. And you see some white area, those areas are their axons. And then this is axons of ganglion cell. These are the axons of ganglion cells which converge, makes the bundle, and becomes optic nerve. Okay. Let's go further. Okay. Further magnification of the retina. So again, you go from choroid, and after the choroid, there is very thin layer of dark layer. This is called pigmented layer. This is the retinal layer where there is a lot of melanocytes, which produced melanin. Then you have nuclei of rods and cones. And these are the axons in the middle. You see the white area, axons of rods and cones here, rods and cones. And then nuclei of bipolar cells, nuclei of ganglion cells, and then axons of ganglion cells again. So in the exam, if I point to any of these, you need to identify. And in the exam, when we put pointer, you see it looks slightly darker. So you have to make sure beforehand you practice it. Same thing here, all the layers again, retina cross section. Those are all the layers. Okay, so this is optic nerve. So when on the posterior side of your eyeball, all the axons of ganglion cells from the optic disc come out, and that becomes optic nerve, which get into your brain to go to your occipital lobe. So earlier I said if we cut cochlea, cochlea is like 3.5 ring, and in every area you're going to see these structures. So you see here, this is one section of cochlea, another section, another section. These all are the section of cochlea. So the superior part is scala vestibuli, scala media, scala tympani. You see, scala vestibuli, scala media, scala tympani. Same thing here. Okay. Let's magnify and see again. So if we focus on one section of the cochlea, you can understand all other section. So scala vestibuli, vestibular membrane, scala media, and outer hair cells, basal membrane, tectorial membrane. This is the ganglion of the vestibular nerve, and then scala tympani. We did it earlier, highly magnified again, scala vestibuli, then you have basilar membrane, sorry, vestibular membrane, basilar membrane, and then tectorial membrane, outer hair cells, and this cavity is scala media. So this scala vestibuli and scala tympani, both are continuous tube in your cochlea. That's why they both contain a fluid called perilymph, and media is in the middle, inner side. So it contains endolymph. Okay. Again, further high magnification of the scala media, vestibular membrane, scala media, tectorial membrane, outer hair cells, basilar membrane. Same thing here again, highly magnified, all the basilar membrane, outer hair cell, and tectorial membrane. This is another cross section of the cochlea, and you can see all the membranes and the cavity and the organ of Corti. This is the taste bud on your tongue, you see. So this is, here is another taste bud, here is another, this is the taste bud. So this is the inner surface of your tongue, this is the superior surface of your tongue. So you see, taste bud has opening on your tongue surface. So when you put sugar here, sugar get inside, and then in this receptors, and then it goes to your brain and you taste the molecules. Okay, so this is all about your lab. Thank you guys.